Assalamu alaikum wa rahmatullahi wa barakatuh. In continuation of our discussion regarding the drugs acting on central nervous system, now our next topic is drugs useful in epilepsy, that is anti-epileptic drugs. For your convenience, we have again further subdivided into four parts. Now this is the first part of our discussion of anti-epileptic. First of all, what is epilepsy? Epilepsy is a chronic disorder of brain function. And this chronic disorder of brain function is actually characterized by recurrent seizures and unpredictable occurrence of the seizure. Now the question is, what is a seizure? Now, a seizure is a transient alteration of behavior of the person, sensation of the person, or consciousness of the person, or there may be mixture of all these three. And this alteration is due to abnormal discharge of cerebral neuron. Now remember, in these anti-epileptics, the choice of medication depends upon the probable type of seizure. Now we will see what are the types of seizures. Types of seizures, first of all, partial. Now, partial seizures may be simple, complex, or initially partial seizures, and later on, or secondarily, they become generalized. In case of simple partial seizure, usually electrical activity is confined to the local area of the brain. In case of complex partial seizure, apart from the above one, there may be involvement of the sensory, motor, or autonomic nervous system, leading to dysfunction of these sides. As far as generalized seizures are concerned, the commonest one are of two, grand mal and petit mal. Grand mal are also known as generalized tonic-clonic seizure, where no doubt loss of consciousness is there, but that loss of consciousness is followed by tonic and then clonic phases of the seizures. As far as the petit mal is concerned, the other name of which is absences, usually there is a brief, abrupt, and self-limiting loss of consciousness, and that lasts only for a few seconds. There may be some other types of seizures also, simple tonic or absolutely atonic, or there may be clonic and myoclonic seizures. And other important types of seizures are in infants, but we call them as infantile spasm or infantile seizures. And the commonest cause in young infants is the hyperpyrexia. Causes of seizure may be known or absolutely unknown. Among the known, there may be some infection like involvement of the meningitis, etc., or encephalitis, or there may be some tumor in the brain, or head injury has occurred, or there may be some toxic or metabolic disorders in the body, for example, hypocalcemia, etc. But the important one is the commonest one is the involvement of the hereditary characteristic, where usually there is defects in the gene, especially which are involved in the voltage gated ion channels or GABA receptor, especially GABA A receptor. Remember, excitation of the cerebral neurons lead to production of epilepsy. And excitation is simply there is excessive discharge and spread of the neuron. There may be some chemicals or the neurotransmitters involved in the brain for these excitation. There may be some excitatory chemicals or inhibitory chemicals. Excitatory includes glutamate, aspartate, homocysteine, acetylcholine, or norepinephrine, where usually the excitatory ions are involved, which are commonly sodium and calcium, while inhibitory types of chemicals and neurotransmitters are GABA, glycine, dopamine, serotonin, etc., where the inhibitory ions involved are chloride ions and potassium ions. This excitation is usually either due to overactivity of the excitatory chemicals or the ions, or decreased activity of the inhibitory chemicals or the inhibitory ions. Like there may be opening of the sodium channel leading to increased positive charges inside the postsynaptic cells, or depressed conductive activity through chloride ions or the potassium ions channels, or there may be involvement of the both. As far as the inhibition is concerned, that may be opposite to that. There may be some increased activity of the inhibitory chemicals or the inhibitory ions, or there may be decreased activity of the excitatory chemicals or excitatory ions. Like there may be opening of the chloride channel leading to hyperpolarization of the membrane, which is a sort of inhibition, or there may be increased conductors of the potassium efflux from the ion, or there may be inhibitory of the sodium channels activity. Let us classify these anti-epileptic first of all according to the molecular targets. Molecular targets, the most important one are the ionic channels, but we call them as voltage gated ion channels. There may be sodium channels, calcium channels, or the potassium channels. In the sodium channels, the important drugs involved are the phenytoin and the carbamazepine, but there may be other ions also related salt to phenytoin and carbamazepine, and some other also lumetragine, glucosamide, and some other drugs. But calcium channels, they are being modified by the ethosuximide and valproate. In bold letter, these are the important one as far as the examination point of view is concerned, or clinical usefulness is concerned. Up till now, only one drug, reticabine, is available which affect through potassium channel. Diagrammatically, postsynaptic sodium receptors, usually these are being affected either through carbamazepine, phenytoin, topiramate, which decrease the intracellular sodium ion concentration, and also through glucosamide, lumetragine, valproate, and zonisamide. While postsynaptic calcium receptors, they are actually mainly affected by valproate or ethosuximide, leading to decrease in intracellular calcium level. Postsynaptic potassium receptors, as I told you, up till now only one drug, reticabine, is available which block the efflux of the potassium, so there is decreased potassium present outside the cell. Second target is the through GABA, increased GABA activity, increased GABA inhibitory effect, either through receptors or through transporter or through the enzyme transaminase involved in the conversion of GABA into its inactive metabolite. As far as the GABA agonist is concerned, there may be phenobarbital or related drug, primidone, or the benzodiazepines are related diazepam, lorazepam, clonazepam, etc. Or there may be some other drugs also. As far as through transporter inhibiting the reuptake of GABA, reticabine is only one drug, and while through transaminase, vigabatrin is the drug involved which inhibit the further conversion of GABA into its inactive metabolite. Diagrammatically, postsynaptic GABA receptors, as we have seen in case of cytohypnotic also, benzodiazepine and barbiturate, etc., they act through the modification of the GABA receptors. So there is decreased influx of the calcium and decreased intracellular calcium level lead to hyperpolarization of the membrane, which is a sort of inhibitory effect. Diagrammatically, GABA neuron is also very important to understand the effects of other drugs, because GABA, as I told you, is converted into inactive metabolite first, intermediate one, that is succinic semialdehyde, and then the remaining metabolites. At the first step, usually two drugs, vigabatrin and valproate, is involved in inhibiting the enzyme transaminase, which convert GABA into succinic semialdehyde, while the valproate also inhibits the second step of conversion of succinic semialdehyde into metabolite through dehydrogenase enzyme, while the tiagabine, that is basically inhibiting the reuptake of the GABA. So that is why leading to more and more concentration of GABA available here in the postsynaptic side to get effect on the GABA receptor. And the next molecular target is the synapse of the glutamate. They inhibit the excitatory glutamate release through two mechanisms. One is through the glutamate vesicle, synaptic vesicle, which is actually synaptic vesicle glycoprotein 2A. Here, levetiracetam is involved, or the related enzyme, this drug is involved, or through one of the subunit of calcium channel, that is A2 delta. Here, gabapentin and pregabalin is important one, along with also other salt also. Now, the next target is again related to glutamate. Now there is glutamate receptors, inotropic type. Two types of receptors are there, AMP receptor and FDA receptors. One drug each, perampanel acting through AMP receptor and felbamate through NMDA receptor. And also lastly, some mixed type of unknown type of targets. There may be felbamate, etc., topamate, zonisamide. But these are the two older ones, acetazolamide and bromide, which were useful in older days as anti-epileptic, and they used to produce their anti-epileptic activity by increasing the carbon dioxide accumulation in the brain. Diagrammatically, postsynaptic AMP receptors, as I told you, perampanel, basically these receptors are involved in influx of both sodium, calcium, but dominantly sodium, little through calcium. And so that is why decrease intracellular, mainly sodium, and very little effect on calcium level. While the NMDA receptors, they were involved almost equally, both sodium and calcium influx, and felbamate used to decrease the intracellular concentration of sodium and calcium through NMDA receptors. So up till now we have discussed the this part of anti-epileptic, and later on we will discuss the remaining part in the second and third and fourth part of the discussion. Up till now, thank you very much.